¿Qué digo? Hola, hola, hola. Buenos días. ¿Todo bien? ¿Sí se escucha? ¿No se escucha?
A ver ahora.
Buenos días. Hola. Buenos días. Hola. Ah, ¿listo? Sí, ya, lo hemos logrado. Thank you.
Welcome everybody to the talk of Laccases, the old enzyme with new application. To be respectful with the speaker, please stay in silence. Dr. Liliana del Carmen Barrera Martinez is a biotechnology engineer with a master's degree and PhD in bioprocesses from the National Polytechnic Institute. She conducted postdoctoral research at the Mexican Petroleum Institute at the Autonomous University of the state of Morelos, focusing on bioethanol production from lignocellulosic residues, energy crops, and other biofuel materials. With 8 years of experience in the biotechnology industry in Mexico and the United States, Dr. Barrera has worked in enzyme and yeast production, in vitro plant cultivation, separation, and purification process, and scaling up biotechnological processes. Her expertise reaches both research institutions and industry applications. Currently, Dr. Barrera is a researcher in the Researchers for Mexico. He's the technical lead of the government research project and collaborates with 3 others. As a level 1 member of Mexico National Institute of Researchers, she has contributed to investigations, attended national and international conferences, and mentored undergraduate, master's, and doctoral students. Please welcome Dr. Liliana Barrera with a round of applause.
Thank you very much for having me here. Thank you very much for the presentation. And well, we're going to start because it's a little late. So the name of my talk today is Laccases, the old enzyme with new applications. Very brief introduction. Everybody knows here what a protease does? Break proteins. Lipases, break lipids. Cellulases, diabases, and laccases. The name of laccases is very special because it doesn't say anything about what it does as enzyme. No? But the laccases was first reported in 1883 in a Japanese lacquer tree. So it's a very old enzyme. This is a laccase from Trametes versicolor. This is one of the most common organisms to produce laccases. Laccases is a benzene-diol oxidoreductase multicopper enzyme. The IC number is 1.10.3.2, and it's an extracellular monomeric glycoprotein. This is a laccase. Well, laccases is also known as a blue oxidase in the enzyme commission nomenclature. Laccases can oxidate a lot of substrates as a phenolic and non-phenolic compounds, aromatic amines, diphenols, and aliphatic amines. Laccases use a molecular oxygen as only electron acceptor, and is very useful for biocatalysis for several biotechnology applications. In the right side for you, we have examples of substrates for laccases. Just take a very close look of these substrates, okay? This is the reaction of the laccases. Don't worry, it's not going to be too much chemistry here. Just, we have water, the oxygen, and water. The laccases, it's from the reducing form to the oxidized form, and the substrate to product, okay? In the bottom part of the slide, we have a reaction of a phenolic compound of laccases. As you can see, we have 4 molecules of this compound. We catalyze the reaction with a laccase, and we lost oxygen to form 2 molecules of water, and we have this substrate oxidate. And then again, with a new laccase, with the same laccase, we have another reaction, the same, oxygen to water. This is a very sustainable reaction. Sources. Laccases are found in so many sources. For example, plants like cabbage, turnip, potatoes, pears, and apples. Insects, like all of those. Bombyx, Califora, Diploptera, Drosophila, Lucilia, Manduca, Musca, Oryctes, Papilio, Fronia, Rhodnius, Sarcophaga, etc., etc. Bacteria, even the bacteria can produce laccases. Escherichia, Streptomyces, Azospirillum, etc. And of course, fungi. You know, Trametes is one of the most common genera that produce laccases. Some others like Pleurotus, Lentinus, etc. Biological roles. Laccases are in everything, so well, almost in everything. What is used to? Lignin degradation, pathogenesis with the insects, detoxification, plants, and morphogenesis and sporulation in fungus. Classification, or all of those enzymes are laccases, but not are the same, you know? Laccases are classified according to the redox potential. Low redox potential are from bacteria, plants, and insects. This is called yellow laccases. High redox potential are from fungi, so they are blue laccases. Applications. As I said before, we have a lot of applications. Biosensor, paper and pulp industry, bioremediation, food industry, textile industry, polymer synthesis, nanoparticles. So they are very useful. But this is what laccases are, where are laccases from. So what are we doing in laccases? I am working in a public center of research. And what are we doing there? Well, this is the same image. We are working with bioremediation, and the new application, it's flavonoid modification or functionalization. This is what are we doing in the lab. And what are we using? Well, we don't care about low redox potential laccases. We use a high redox potential producing via fungi. The first case of a study is flavonoid modification, a quercetin. Quercetin is a very common flavonoid in the nature sources. I guess many of you have a very nice breakfast with fruit. Watermelon, melon, maybe coffee. Yeah, well, quercetin is there. Maybe you have some chilaquiles with onion. Onion has quercetin. Quercetin is that molecule, that compound with 3 rings. 2 of them are phenolic rings, and there's in all that vegetables, fruits, as an apple, olives, onion, even in wine, beer, we have quercetin. Health benefits of quercetin. As a flavonoid, they have a lot of health benefits. Anti-age, you can eat quercetin in order to get younger. For cardiac illness, it's a very potential antioxidant. It can help with allergies. If some of you have some allergies, you can eat quercetin. And one of the most important is cancer. Quercetin can help to reduce the chances to have cancer. Now, what is the problem? Quercetin is a very nice compound that helps us a lot in our health, but what is the problem with the quercetin? The quercetin, as many of those molecules, have the problem of the bioavailability. What is that? Well, if you take a pill for a headache, the bioavailability of this pill is that get in the right place and do the right thing, This is not the case of quercetin. Quercetin have a very, very, very poor solubility in water, have problems to go through the membrane of the cells, and have a very, very, very high rate of clearance in our body. That means quercetin is very good, but not for us, at least not in that form. Well, then we have to do it something. Do you remember that I said Laccases have a lot of substrates to work? One of those is phenolic compound. Quercetin is a phenolic compound. So we use a quercetin from Trametes sanguineus to modify this compound. What is a Trametes sanguineus? Trametes sanguineus is a water fungus with a very nice lignocellulosic enzyme complex. This is a representation of Trametes sanguineus. So we have quercetin, we have our fungus. So what next? This is the work of Carlos, a student have been working in this project. So we produce the enzyme with the seed cultures with bran flakes media. Yes, bran flakes for our breakfast. Then with this seed, we inoculated the production culture that have bits of pine sawdust and with straw, the lignocellulosic residues. And after 7 days, we check the enzyme activity. Well, now we have our enzyme, our crude enzyme extract. So we try with these parameters to modify quercetin. We try with 3 pH, 3, 4.5, and 5.7 phosphate and acetate. This is the most the optimum conditions for the enzyme. Acetate buffers 4.5, and the quercetin was 10 millimolar. Enzyme was 0.8 units per mil of the reaction, 40 degrees, 1.5 RPMs, and 24 hours of the reaction. And these are the results, okay? As you can see in the picture, we got different colors of the reaction. And what is this? Well, this is with no enzyme. This is quercetin, just quercetin with buffer, and that's it. This is pH 3, and the color starts changing. This is pH 4.5, and the last one is pH 5.6. So we did something with the quercetin. Well, not us, Laccases. But what else? One of our tools to see if something happened with the molecules is the UV-Vis scanning. It's a quickly and cheap way to see if we are doing something with this molecule, with these compounds, So the BE here it's with non-enzyme. This is the fingerprint of quercetin in a UVB scanning. And this is for the 3 pHs. 3 is the green one, 4.5, the red one, and 5.6, the blue one, you know? So yes, this graphic told us that we did something with the quercetin. It's not more quercetin, it's quercetin modified. What? Until this point, we didn't know. We just know that we did something with this quercetin. Well, then we decide to keep forward. So we choose the 5.6 pH because it's, if you can see, is the most different profile from quercetin. So we choose that. And we escalate the reaction to a chemical reactor. We put there quercetin, crude enzyme, buffer, 40 degrees, the same speed agitation, and the same time of reaction. And then what? Well, this is the reaction, the same color, and the fingerprint again of our product. Please forget about lavado 1, lavado 2, it doesn't matter, because we just work with sobrenadante. We centrifugate all this, we recover the sobrenadante, and is the red one. So also in our chemical reactor, we did something with the quercetin, you know? This is another tool to see what's going on with our reaction. This is a HPLC chromatographer. Quercetin is here. This is just quercetin. From the bottle of Sigma, we took quercetin, and injected in an HPLC, and we obtained this. This profile. Then our reaction product that from here to the end, we are going to call it modified quercetin, is this one. As you can see, quercetin is not anymore. Well, just a little bit. You can see that? Yes? Sure? Okay. So, yeah, we did it. We modified quercetin. And then what? Well, our strategy to see if the bioavailability of quercetin increases or not has to test this one, the modified quercetin, in cell lines. We work with 2 cell lines. If you remember at the beginning, I told you the quercetin is one of the most famous flavonoids to treat or to reduce cancer chance, to have cancer. So we work with that, with a healthy cell lines called Vero and cervical cancer line called Gila. So we work with both to see if our quercetin was better or not, modified quercetin. And this work was did it by la maestra Flor in Ciatecto, but she is an expert in cell lines cultivation and all of those stuffs. In this plate, we can see the cell cultures with quercetin and no quercetin. Well, one of the most wonderful, wonderful results is this one. We check the viable cells using a cytometer. So this is with quercetin. As you can see, quercetin. Don't do that with the Gila cells, cervical cancer cells. You can put even 200 micromolar quercetin, and you got maybe a 90% of cell lives. So maybe this indicated that the cancer cells with normal quercetin still alive. But with the modified quercetin, with our quercetin, in 200 micromolar concentration, we got maybe the half of the viable cells. just around 50% of the variability. So yeah, we did it. At least at this point of the research, we can say that our quercetin, it's better to kill HeLa cells than quercetin normal. Okay? Questions? No? Okay. But what about the healthy lines, the healthy cells? If you know, maybe all of the drugs to treat cancer, the problem is that it can kill cell cancers, but the same drugs can kill healthy cells. So this is a healthy line, pero, and in 200 micromolar, that was our concentration of our quercetin, killed just a half of the cell of the cancer cells. Now here, killed less than 80% of cells. So there, our modified quercetin can kill cancer cells, but no healthy cells. So that's a very good news. And the conclusions until now is that it was possible to modify quercetin using a crude enzyme extract. At least 2 products of reaction were formed, as you can see in the HPLC profile. Modified quercetin, that's another thing, was more soluble in water than quercetin. In the picture that I show you in the photo, that it's from quercetin to quercetin with phosphates 5.6, the quercetin was more soluble, so in water. And apparently, the viability of modified quercetin improved compared to the quercetin that showed the cell viability assay with Vero and HeLa cell lines. So until now, we know that. We have to work harder to realize what are the products formed. We are working with HPLC mass and infrared spectroscopy, and that's it. To see what are our products. This is a project supported by CONACYT, and this is the nice group of researchers and students that work with this project. And all this is just thanks to them. And the other use that we are doing with these Laccases is bioremediation. You know? This is the second case. We are, I am not from Jalisco, from Mexico City, but I am living in Jalisco. And I am working with research. And I have a nice enzyme, you know, called Laccases. Now, in Jalisco, we produce, well, they produce tequila, we consume tequila, but tequila produces 2 residues. One is agave bagasse, that everyone wants agave bagasse, everyone works with agave bagasse. Now, the second residue is vinage. Vinage, tequila vinage, is maybe nobody knows about that here. Tequila vinage is a bioproduct of agave fermentation. It's a wastewater, dark color, complex composition, high water, alcohol, sugar, phenolic compounds, and proteins. Yeah, it have a very, very low pH, and it's a very, very hard problem for tequila. Why? Because 1 liter of tequila produces between 10 and 30 liters of vinage. And this is the uses that persons gave to vinage. They uses to irrigate the fields. So the annual estimation of production is 7,000 millions of liters of vinages. So we have to do something with that. We propose a phytotoxicity study. This is a work we are doing with Doctora Soledad and Doctora Verónica. Well, as you know, I hope so, Laccases work with phenolic compounds. Vinasas has phenolic compounds. And then we said, well, let's try to do something with vinasas and our Trametes. Trametes sanguineus is the king of my life. So we start working with vinasas at 50% in a bioreactor. Yep, we have the same strategy. We have a seed culture. With the seed culture, we inoculate the bioreactor, and then after 5 days, we measure color, total phenolic compounds, conductivity, and total sugars, and chemistry, oxygen demand. And then we try with these vinases to irrigate plants, tomato and lettuce plant. We measure the chlorophyll, height, fresh weight, dry weight of the area part, this part of the plant, and the root, leg, fresh weight, and dry weight, And let's see. This is all the results. This is vinasas at the beginning. As you can see, it's a brown color, dark brown. After 5 days with our Trametes, the color was lighter. It's just more like a brown, light brown. So Trametes can decolorize the vinasas. They use phenolic compounds to grow and produce laccases. So until now, as you can see, just to see the vinasse, we know that we did something with that. Then we measure the laccase production in the reactor. So we try, no, this is not in the reactor, this is a flask. We try with vinasas at 50%, 70%, and 100%. I mean, just vinasas. And this is the laccase activity, volumetric laccase activity in 10 days. As you can see in this graphic, the laccase production was very high, more than 1,500 units per liter. So we can decolorize vinasas, we can produce laccases, and then we measure these phytochemical characteristics of vinases with vinasa without the treatment and the treatment at the different concentration. Yeah. So let's see phenolic compounds. In all the cases, the phenolic compounds just get low if you compare the vinases without treatment and with treatment in all the concentrations. It was the same with the total sugar content. In all the concentration, we have a diminution of this concentration. The chemical oxygen demand, the same, it gets low. So we presume until now that vinasas was less toxic than at the beginning. So we use these vinasas to irrigate the plants. This is control, it means water. This is, here we use vinases at 50% treated. And this is tomato plants. It looks quite the same, but the roots. And this is lettuce plants. Here with the vinases, we have small plant, but the same roots, the same height, the same weight, fresh and dry. So yes, we can detoxify vinases, use it to irrigate some plants, some culture. But the problem here is the aerial part of the plant is not the same. With the control, with water and vinasse. Here is the same. As you can see, this plant looks healthier than this one. So we have to do something else. And here is not the what are we doing or what are we doing now, but something else we can do. We are trying to accumulate this process, the tramete sanguineous process, with a wetland strategy to get the vinasse with less toxicity for the plants. And tequila vinasses can be a cost-effective and available culture media for lacase production. As far as we know, this is the first report that used vinasses to produce lacases from Trametes sanguineus. Trametes sanguineus contribute to tequila vinasses bioremediation by reducing the color, phenolic content, and decrease in phytotoxicity. And reusing treated vinasses appears a promise in the early stage cultivation for economical important vegetable seedling, such as lettuce and tomato. And please, now this is to dedicate this presentation to all the women that have to be with me in my journey until now. Of course, my mother, my grandmother, my mother-in-law, and all my friends, my colleagues that have been with me to be here. So, of course, all the women in this room, thank you very much for having me. And that's all for me.
Have questions, please raise your hand. Okay, to start with.
Hello. First of all, thank you for your presentation. It was very, very interesting. And I wanted to ask in the health part of the research of the cancer cells, you already showed that there was like no change in the availability of the cells in the treatment with
Modified quercetin or quercetin?
with the modified,
Uh-huh.
like the with the viable cells, like the healthy,
Uh-huh.
like the strain of the cells. And has there been any other side effects that these cells present when treated with modified quercetin?
On July, we don't know. I present just the viability test, that is cytotoxicity test. There's another test that we have to do it with this, and even in with rats or mice, to see if there's another side effects or not. As you can see, we just try with cancer cells and healthy cell line to see if the healthy cell line suffers some kind of damage. But until now, you can say no. But if we're modified quercetin will have side effects, I don't know. I hope no, but I cannot say anything about that now. Right now, we have more research. You are very welcome.
Hi.
Hi.
When same talking about the cancer cells, I was curious, like, what kind of cells did you test it on, and if you would have like other results in different type of cells, like from different tissues.
Yeah. Yes, the answer is yes. We have been working with breast cancer, colon cancer, and leukemia, but this is just the trabajo of the work of Carlos. So Carlos worked with cervical cancer cells. Now, one student, my PhD student, David, is working with breast cancer and colon cancer. And colleague from the University of Guadalajara, they are working with kidney cancer. Yeah, no, liver cancer cell line. So we are trying to test as many cell lines as we can, but it's very expensive and we take a lot of time. So we are working in that. And this in this year, we are working with leukemia cell lines.
Thank you.
You're welcome.
Could one of the objectives of the modified care routine
Mhm.
be for it to be available for the daily day-to-day health benefits for like the everyone
Mhm.
to consume it?
Yes. The original title of the project is a very long title, but we are interested in treat the most common illness of the Mexican population, like cancer, obesity, diabetes, cardiac issues. Yeah, the answer is yes. But now we start with cancer because that's the cell lines that we have at the center. I am looking for some other collaboration to work with diabetes and obesity. I'm working on that.
Hello, first of all, thank you for your presentation. It was very interesting.
Thank you.
And well, you showed some images of the HPLC.
Mhm.
Well, do you have any guests from the HPLC of what compounds do you have? Or are you planning to perform a NMR or something like that?
Yeah. Well, the HPLC, I show you the UVB scanning and HPLC because we have those in the lab. This is our tools that we have just around the corner. But yes, we are doing now UPLC mass and spectroscopy to elucidate the structure of the molecules. Yeah, we are doing. But this is in some other labs, in some other states, even the HPLC mass is in Yucatan, and spectroscopy is in Mexico City. Well, it's kind of difficult to get the results as fast as I want. But yes, we are doing that.
Great,
Thank you.
you.
I didn't say anything. I didn't say anything yet.
You are gonna ask a question, so you please speak loud.
Well, first of all, thank you for your talk. I also thought it was very interesting. And what I wanted to ask you is more related with the bioremediation part. Do you know if there are any like long-term effects on the soil that the plants are?
Well, no, we don't have about our our vinas or our vinas.
Like the soil that they.
Well, the people there in Jalisco use this residue to irrigate the fields. Now the result is acidification of the soil. They have a lot of minerals, so it gets some minerals in the soil. So after some years, the soil is it doesn't work anymore to get any culture. So this is, and we hope with our work, the people can use these vinazos to irrigate the fields, but without side effects.
Hello. I have a couple of questions. First of all, your lab, well, this research seems to be very familiar with the use of Trametes sanguineus, and I wanted to ask, did you guys have to develop your own culture methods for this? Were these found in literature? What was the process? Because I know that these are like very long known, very for long known funguses and used medicinally in East Asian cultures, for example, but they're not much heard of here. So how did you like get so familiar with it? What and how did you guys work with it?
Well, I was working with Trametes sanguineus from 2016, maybe almost 10 years in my postdoctoral research. I started using Trametes sanguineus. Trametes sanguineus is a very nice red fungus. And well, it was hard at the beginning because it doesn't want to produce laccases. So
Fungos are difficult.
it's yeah, it was terrible to me because it was my research. So after trying so many media with some residues from agave bagas, sugarcane bagas, this is rice residues too, we found finally this sawdust pine and the wheat straw, the combination of both these, it gave us a very nice results. And we use copper sulfate as endosser to get Laccases. And well, after 10 years, we got this media. The conditions are 28 centigrade temperature and a very low speed agitation because Trametes sanguineus doesn't like to be as a yeast or a bacteria. And that's it. Yeah. At the beginning was very hard, but now everything is cool with Trametes sanguineus.
I can ask another question. Yeah, and also your results on tests with the HeLa strongly suggest that there is selective induction of apoptosis on the HeLa culture cell lines. Have you thought about doing protein assays to find which, I guess, pathways are activated in this regard? Because I think there's a lot of research that can be done and cross comparing how is the apoptosis induced, because that could give a lot of insight into what molecule you're producing and how it works, and if it has any considerable interactions with other cultures.
Well, yes, we are doing, this is just one study of the cell lines, but we have some others, and now we are working with that. We are doing, I don't know how to say in English, ensayo cometa, to see how the cells die, and some others. And the students for my PhD students is working with that kind of information to see what genes are just like affected with modified quercetin, and see what the proteins are affected with the modified quercetin. And even there's a lot of information about what is going on with quercetin and cell lines. We already know what genes are affected with this quercetin. So we are working on that.
Thank you very much.
You are very welcome.
Thanks. We're going to pass with the two last questions. If you have any further questions, you can reach the doctor.
Hi, I wanted to ask, can the process such as the one that you use for the tequila vinases be used for other beverages, such as beer, for example, or another one?
Well, Laccases are used to, in the food industry, one of the uses is to clarify wine, clarify beer, clarify juices. So we can use it in that way to clarify this beverage, or we can use it to decrease the toxicity of the residues. But just remember, we need some kind of substrate that like Laccases, like phenolic compounds or not phenolic compounds, but you have to, the residue have to have something like that to produce Laccases, and Trametes sanguineus can grow.
Is there like a minimum amount for phenolic compound that should be in the beverage so we
Yes.
can start using the Laccases?
Yes, the answer is yes. We are trying with some, I don't know the number because we are not measured that, but I can say yes because we are working with different vinases from tequila's industry, and we already know that vinazas is not as dark as you can see in this picture. Laccases is not working. Trametes is not growing, and Laccases are not producing. So they need a very, very, very dirty vinazas to grow and produce.
Thank you.
You're welcome.
Hello, thank you for the talk. It was very interesting. And my question is like, I understand you had to optimize the process in order to obtain as much Laccases as you could from the Trametes sanguinea. Right now, what percentage is the yield around?
Oh, well. I have to calculate the yield.
Yeah.
But I can say if we compare our production from other authors in the literature, we are high.
Okay. Thank you.
Yeah. Yeah, very much.
So just very fast, a question. Is Prometheus used in the collection to the name for all mycology class? Is the strain Prometheus within the collection you made?
Yes,
You just
please be careful with that. Yes, I have. I gave to Dr. Jose Luis a Trametes sanguineus, my Trametes sanguineus. So please be kind with it. It's a very, very nice fungo. I love it. I love it. So, yeah, is there? You're very welcome.
Thanks a lot for your talk, for your presentation, for the knowledge shared with us. Here is a little present for the STEM community and our sponsors. Thanks a lot, Dr. Ibar.
No problem. Thank you very much. Thank you very much, everyone. Thank you very much for having me. No te preocupes. Claro.
Por los errores técnicos.
Buenos días todavía.
Hola, buenos días.
Ahorita estamos terminando la plática anterior. ¿Tiene su presentación lista?
Es parte.
¿Cómo disculpa?
¿Puedo compartir?
Sí, administro los permisos.
Muchas gracias.
Porque creo que... Ay, sí.
No se parece a la
A ver.
O igual también puede ser.
Ahí va.
Hay que agradecer,
A ver. Sí, sí, aquí igual, ahorita a las 11. A las 11. A ver, doctor, creo que ya pude compartir.
¿Me avisas si la ves, por favor?
Claro que sí.
Ya debe estar.
Ya,
Perfecto.
perfecto. Y no tardamos en comenzar.
Muchas gracias.
Muchas gracias por nos dar un poco de su tiempo y estar aquí presente.
No, al contrario, muchas